Hello dear students, Namaskar. Now we are going to discuss about citric acid cycle. That is, it has got other names, that is tricarboxylic acid cycle and Krebs cycle. So these are the three names given to this cycle. There is one citric acid cycle, two tricarboxylic acid cycle, three Krebs cycle. Is there any significance for this? Why they are calling it citric acid cycle? Because so many other acids are participating in this cycle, but they are giving the name citric acids for this cycle. Because citric acid is the first acid which starts this cycle. So from citric acid the cycle originates, or the cycle is initiated with citric acid. That is why it is given citric acid cycle. Another name is tricarboxylic acid cycle. Why it is tricarboxylic? Because all the acids which are participating in this cycle are having three carboxylic units. They are all three carboxylic units carrying acids. That is why they have given the name tricarboxylic acid cycle. Otherwise, citric acid itself is having three carboxyl groups. So it is tricarboxylic acid cycle. Another name given to it is Krebs cycle. Why it is Krebs cycle? Because Krebs is the scientist who discovered the cycle and who organized the cycle, who formulated the sequences of the cycle and explained the significance of the cycle. So in the name of that great scientist, the cycle has got the name Krebs cycle. So this is a very, very important cycle in our body because this cycle integrates all the three metabolic pathways. All the three metabolic pathways of carbohydrate, protein and fat. So the energy which are coming through the carbohydrate energy line pathway, the energy which are coming through the protein line pathway, and the energy which come through the fat line pathway, all these are integrated in the citric acid cycle. So why they are integrating these three pathways? What is the significance of this integration? The significance is this, because we are taking carbohydrate, we are taking fat, we are taking protein. Why we are taking? Why we are taking carbohydrate? Why we are taking protein? Why fat? Because we are utilizing the energy which are present in the carbohydrate. We are utilizing the energy which are present in the protein and the energy which is present in the fat. So these energies are captured to the cellular functions. This energy is captured for performing the cellular functions. We have to do through some mechanisms. That mechanism is citric acid cycle. So how energy is present in carbohydrate? From where this energy came? From where the energy came to fat? The energy which are present in this dietary energy lines are nothing but solar energy, the energy of the sun. At what time this energy was installed into these biological molecules of carbohydrate, protein and fat? At what time? It was installed at the time of photosynthesis. So this photosynthetic solar energy which are present in carbohydrate, protein and fat are derived out through citric acid cycle. That is why this cycle is having importance, biological importance. Now, how energy is installed into these biomolecules? Because the photosynthesis in the presence of chlorophyll, the water will split into oxygen and hydrogen, and that hydrogen will be utilized and the oxygen will be driven out. So this hydrogen, which are carrying electrons. So in that electrons, this solar energy is installed by the sun. And with that solar energy, these carbohydrates, protein and fat, all these are formed, which are mainly synthesized, by plants. Animals are not able to synthesize energy like plants. So this energy is traveling through food, and that enters in our body. When it enters in our metabolic system, we will split these compounds. We will split the carbohydrates into starch, glucose, and finally pyruvate after this aerobic glycolysis. So carbohydrate is oxidized to starch, glucose, and pyruvate. And protein is disintegrated from dipeptides, tripeptides, polypeptides into small amino acid units, acidic amino acids, basic amino acids, sulfur containing amino acids, like that, various amino acid basic units. And in case of fat, that is disintegrated into acetyl-CoA semi-molecules. So the end product of metabolic pathways of these three energy derivative: carbohydrate, pyruvate, protein, amino acid, fat, acetyl-CoA semi. So in these three forms, that is in the form of pyruvate, in the form of acetyl-CoA semi, in the form of this acetyl-CoA semi, pyruvate, and amino acids, this energy molecules, diet molecules, enters into citric acid cycle. Okay, now what happens after citric acid cycle? So through citric acid cycle, is an acidic cycle, so many reducing activities takes place, and reducing compounds are formed like NADH2, FADH2. These are this solar hydrogen electron carrying compounds. We can call them like that. Solar electron energy carrying compounds. NADH2, FADH2. From one molecule of NADH2, we get 3 ATP molecules. From one molecule of FADH2, we get 2 ATP molecules. So they are representatives of energy. So by this cyclical process of various transformations of this, various sequential transformations of these various tricarboxylic acids, the energy present in those electrons or those hydrogen atoms are captured in the form of NADH2 and FADH2, and they are channeled to the electron transport chain. So in the process of electron transport chain reactions, this energy will be captured in the form of ATP. From one molecule of NADH2, 3 ATP molecules will be formed. One molecule of FADH2, 2 ATP molecules are formed, roughly. Now they are saying that it is less than. Instead of 3, only 2.5. Instead of 2, only 1.5. Leave it. But for convenience, we are believing that one molecule of NADH2 represents 3 ATP. One molecule of FADH2 represents 2 ATP molecules. Okay. This energy is captured in the form of ATP. And that ATP is known as a cellular currency. So that ATP is very much palatable for the cellular activities. I am talking to you, you are listening to me, we are doing all these activities, our blood is flowing in our vessels, our heart is beating, our kidney is functioning, our brain is functioning, everything is being done with this energy of ATP. So the ATP is generated from this reducing equivalents, NADH2, FADH2, from citric acid cycle. Then that is channeled to the electron transport chain. And finally what happens? The extra electrons which are formed will be captured by oxygen. And two electron carrying hydrogen atoms will be combined with one molecule of one atom of oxygen to form water. So at the end, otherwise, if we are not oxygenating the cells, what will happen? The cells will be burned by this hydrogen. So to protect the cells, we are protecting the cells. We are taking oxygen in the form of respiration. So that respiratory oxygen will neutralize these electrons to form water. Suppose we are not respirating, what will happen? We will die. Why we are dying? We will be killed by these hydrogens. Who is actually killing us? We are being killed by hydrogen. If we are not respirating, who is killing us? Hydrogen is killing us. So neutralize this hydrogen, we need oxygen. That's why we are taking breath. Okay, so this is the story. So what is the whole energy process? It's a story of energy, how the energy traveling in nature, from nature to living beings, how energy is traveling. This is the story of energy. That story begins with splitting of water into hydrogen and oxygen. And oxygen is expelled out and hydrogen is accepted. And end, what happens? This hydrogen is accepted again by the oxygen and forming water. So the story of energy begins with splitting of water into hydrogen and oxygen. The story of energy ends with formation of hydrogen and oxygen and formation of water. So in the beginning, water is split into hydrogen and oxygen. In the end, water is formed by the combination of oxygen and hydrogen. This is the story of energy. Now, we will discuss about what is citric acid cycle. In the exam point of view, what is the importance of citric acid cycle? Usually, essay questions are being asked from citric acid cycle area. Usually the essay question is like this. Describe the various steps of citric acid cycle with the help of a systematic diagram. Explain the various steps of citric acid cycle with the help of a diagram. Mentioning 1. The sites of energy production. 2. The sites of inhibition. 3. The sites of regulation. So we have to explain the citric acid cycle by mentioning the sites of energy production, the sites of inhibition, and the sites of enzyme regulations. So this is how usually the questions are being asked. So no doubt you have to draw this diagram of the citric acid cycle. In all the textbooks, it's beautifully given. You have to draw it several times and make it by heart. No, there is no other way other than that. Because that question is usually being asked as university question paper. Okay. Now, I'll just tell, there are 10 compounds who are participating in this citric acid cycle. There are 8 enzymes who are participating in this citric acid cycle. There are 3 sites of energy production, that is oxidative phosphorylation. One is substrate level phosphorylation. The total 4 sites of energy production. Okay. There are 3 sites of enzyme regulation. There are 3 sites of inhibition. This is citric acid cycle. Okay. Once again, for you to remember, I'll just say there are 10 compounds. 10 compounds who are participating in citric acid cycle. 8 enzymes which are participating in citric acid cycle. 4 sites of energy production. 3 sites of inhibition. 3 sites of regulation. Finish. So, I'll tell the compounds which are participating in citric acid cycle. The first compound is citrate. second compound is cis-aconitate. Third compound, isocitrate. Fourth compound, oxalosuccinate. Fifth compound, alpha-ketoglutarate. Sixth compound, succinyl coenzyme A. Seventh compound, succinate. Eighth compound, fumarate. Ninth compound, malate. 10th compound, oxaloacetate. Finish. These are the 10 compounds which are participating in this sequence of reactions. Among that, first two reactions, there is formation of citrate to isocitrate is guarded by aconitase enzyme. Aconitase enzyme. Then next reaction, that is oxalosuccinate to alpha-ketoglutarate, that is succinate dehydrogenase enzyme. Okay. Then next is alpha-ketoglutarate enzyme. That alpha-ketoglutarate is converted to succinyl coenzyme A, alpha-ketoglutarate dehydrogenase enzyme. The succinyl coenzyme A to succinate is by succinate thiokinase. Then succinate to fumarate, succinate dehydrogenase. Fumarate to malate, fumarase enzyme. And malate to oxaloacetate is malate dehydrogenase. Then oxaloacetate to citrate is citrate synthase. So these are the 8 enzymes. Then which are the energy producing sites? This is very important. Energy producing sites are, the first one is isocitrate dehydrogenase. There one molecule of NADPH2 is formed. The second energy site is alpha-ketoglutarate dehydrogenase. There one molecule of NADPH2 is formed. Then next is succinate dehydrogenase. There one molecule of FADH2 is formed. The next is malate dehydrogenase. There one molecule of NADH2 is formed. Then at another site, that is succinate thiokinase, that conversion of succinyl coenzyme A to succinate. There one molecule of GTP is formed. That GTP is substrate level phosphorylation. But that also gives one molecule of ATP. So total how many ATP? 3 NADPH2 are formed. NADH2 are formed. Not NADPH2. NADH2 are formed. 3 NADH2. So one molecule of NADH2 gives 3. So 3 into 3, 9. And at one side FADH2 is formed. From FADH2 we get 2. So 9 plus 2, 11. Then from GTP we get what? 1. So 11 plus 1, 12. So from one molecule of glucose, you know the glycolysis, 2 molecules of pyruvate are formed. So we have to double this. So that total 12 plus 12, that is 24. 24 ATP. And from aerobic glycolysis we get 8. So 24 plus 8, 32. So from single molecule of glucose we get 32 molecules of ATP by the process of this citric acid cycle and electron transport chain. Okay, that is the energetics of this pathway. Then inhibition. Inhibition, what are the inhibiting compounds of citric acid cycle? One is fluoroacetate, second is arsenate, third is malonate. So these are the compounds which inhibit. So there are inhibition sites, very important. This fluoroacetate, that is inhibiting the aconitase enzyme. And aconitase enzyme is inhibited by fluoroacetate. The arsenate inhibits alpha-ketoglutarate. Alpha-ketoglutarate is inhibited by arsenate. Then malonate inhibits the succinate dehydrogenase. So succinate dehydrogenase is inhibited by malonate, malonic acid. So these are the 3 inhibiting compounds. Then 3 regulation sites. All pathways, the first reaction is always a regulation steps. So citrate synthase enzyme is a regulatory enzyme. Then we have isocitrate dehydrogenase enzyme is another regulatory enzyme. Then alpha-ketoglutarate dehydrogenase, that is also another regulatory enzymes. So these are the three regulating enzymes. That is citrate synthetase enzyme, then isocitrate dehydrogenase enzyme, and alpha-ketoglutarate dehydrogenase enzyme. So these are the three enzymes which are regulated. Now, next important peculiarity of this cycle is, so we have seen the sequence of reaction, the energy production sites, the inhibitory sites, and the regulation sites that we have seen. Now we can discuss about the significance and the uses of this cycle. Significance and the uses of the cycle. So when we discuss that, the first thing, first in significance, it integrates the three metabolic pathways. That is the most important significance. It integrates the protein metabolism and carbohydrate metabolism and fat metabolism into a single cycle. So it's the most important significance. Okay. Second significance, by this cyclical process, it generates reducing equivalents or reducing compounds like NADH2, FADH2, and that those things can be channeled to the electron transport chain. So the reducing elements are generated by this cycle. Next significance, this cycle is important for its amphibolic activity. Amphibolic, that word is very important. Amphibolic. What do you mean by amphibolic? Amphibolic means both anabolic and catabolic. So it has a property of anabolism, it has a property of catabolism. Anabolism means creative, catabolism means destructive. So it is a cycle which is having a creative aspect, at the same time it is having a destructive aspect. That is the amphibolic character of citric acid cycle. Next significance is it is anaplerotic. Anaplerotic. These words are very important. When you write in exam, you have to mention these words. Amphibolic. You write in capital letter amphibolic, underline. You will get good marks. Then anaplerotic. Anaplerotic is nothing but replenishment of its own ingredients by its own way. That is, for example, pyruvate can be replenished with oxaloacetate. Malate can be replenished. Glutamate, from glutamate and alpha-ketoglutarate, they can be replenished. So, any deficiency of its ingredients at any time, at any point of time happens, that will be immediately replenished. So, that is an anaplerotic activity of this citric acid cycle. I told you this word amphibolic and anaplerotic are important. So you have to mention that word itself. There is no other word you can use, alternative words. No other words. Amphibolic means amphibolic. So what is amphibolic? So that we have to discuss. Amphibolic is both a combination of anabolic activity and catabolic activity. So let us first discuss about the catabolic activity of citric acid cycle. So how these compounds are channeled into the citric acid cycle, how they are utilized, how they are catabolized, that is very important. That is how we have certain important channels, so certain compounds which are present in the citric acid cycles act as the channels through which this metabolic world enters into this citric acid cycle. The first is, of course, it is oxaloacetate and acetyl coenzyme A. Oxaloacetate is a very wonderful compound. It can be converted so many things into. So for example, aspartate is converted to oxaloacetate, and pyruvate can be converted to oxaloacetate. So oxaloacetate is very important. It's one of the important channels. So through the channel of oxaloacetate, pyruvate is mainly entering. Pyruvate is also a versatile compound because pyruvate is a very important keto acid which act as a transaminating keto acid. So many amino acids can be transaminated into pyruvate, and that pyruvate can enter into this citric acid cycle. Regarding carbohydrate metabolism, this glucose undergo aerobic glycolysis, aerobic oxidation into pyruvate, and that pyruvate enters into citric acid. Then for another example, amino acids can be converted to pyruvate. For example, glycine, serine, cysteine, all these can be converted to pyruvate, and through pyruvate they will enter into citric acid cycle. Another group, tryptophan converted into alanine, and alanine is transaminated to pyruvate and enters into citric acid cycle. So pyruvate is an important pyruvate, then to oxaloacetate, and so many compounds are entering into the citric acid cycle. And of course, acetyl coenzyme A, the fat metabolism, is a representative of fat metabolism. And acetyl coenzyme A is formed by the beta oxidation of fatty acids, and that enters into the citric acid. And at the gate of alpha ketoglutarate, glutamic acid enters into the citric acid cycle by transamination. Glutamic acid can be converted to alpha ketoglutarate by the transamination. And citric acid cycle is actually a acidic cycle, so it won't accept any basic amino acids. Directly, it won't accept any basic amino acid. So these basic amino acids have to be converted. So the basic amino acids like arginine, histidine, these can be converted to glutamate. Then glutamate transaminated to alpha ketogluteric acid and enters into the cycle. So indirect way of entrance of the basic amino acid. Along with that, proline can also be converted to glutamate and then to alpha ketoglutarate and enter the cycle. And another important gate is succinyl coenzyme A gate. So succinyl coenzyme A, through succinyl coenzyme A gate, all the odd chain fatty acids are converted to propionyl coenzyme A, and this propionyl coenzyme A will be converted to succinyl coenzyme A and enters into the citric acid cycle. At the same time, certain compounds amino acids will be converted to propionyl coenzyme A. For example, valine, isoleucine, methionine. These are converted to propionyl coenzyme A, and that converted to succinyl coenzyme A and enters into the cycle. At the same time, fumarate. At the level of fumarate, this phenylalanine and tyrosine, both can be converted to fumarate and they enter into the citric acid cycle. And of course, aspartate is converted to oxaloacetate and enters into the citric acid cycle. So this versatile, different kinds of fat metabolism, protein metabolism, carbohydrate metabolism, all those compounds, end products, are entering into the citric acid cycle through these various gates. So important channel gates are, one is oxaloacetate, through acetyl coenzyme A, through alpha ketoglutarate, through succinyl coenzyme A, through fumarate, oxaloacetate. So like that, these compounds are entering into the citric acid cycle. So this is a diagram is given here. You have to draw that diagram. So if you are drawing that diagram, everything is explained. Everything is explained. Now, this is the catabolic aspect. Regarding anabolic activity, so certain compounds can be formed from citric acid cycle. So see, for example, this oxaloacetate and pyruvate, they are interconvertible. Pyruvate carboxylase enzyme is used to convert the oxaloacetate to pyruvate to oxaloacetate. Like that, glutamate can be converted by transamination. The alpha ketoglutarate can be converted to glutamic acid, and that glutamic acid can be utilized for synthesis of gamma aminobutyric acid, GABA. So that is an anabolic. So GABA, the synthesis of GABA is having a very important role played by this citric acid cycle. Succinyl coenzyme here, that's which plays a very important role in the synthesis of heme. So in the heme, the initial reaction, glycine, succinyl coenzyme formation, the heme synthesis starts with the succinyl coenzyme. So heme, the synthesis of heme, citric acid cycle is having a very important anabolic role. At the same time, the process of ketolysis, this succinyl coenzyme is used. Okay. Then malate is having a very important role in the gluconeogenesis. So these are this picture will explain everything. These are the various anabolic roles. So both the anabolic and catabolic together we call it as amphibolic. And I told the anaplerotic. Anaplerotic is nothing but a replenishment of the various components of the citric acid by interconversion of various compounds. For example, pyruvate, oxaloacetate, glutamic acid, alpha-ketoglutarate, such interconversion and replenishment. So these are the important aspects of citric acid cycle. So one thing I forgot to mention, there are as this reducing equivalents are also formed from the citric acid cycle, carbon dioxide is also released from the citric acid cycle. So which are the sites of carbon dioxide release? One is isocitrate dehydrogenase enzyme site. The other one is alpha-ketoglutarate dehydrogenase enzyme site. At these two sites, carbon dioxide is also released from citric acid cycle. So that also very important, you have to mention while you are drawing the diagram of the citric acid cycle. So far important aspects of the citric acid cycle we have discussed. So I'll just narrate the whole thing. So when you come to citric acid cycle, if an essay question is being asked, just focus on this important aspect. First, the general aspect of the citric acid cycle. What is the significance of citric acid cycle? It is the integration of 3 metabolic pathways. Okay, then we come to the citric acid cycle. First thing, the sequence of citric acid cycle. Second thing, the sites of energy production. Third thing, the sites of inhibition. Fourth thing, the sites of regulation. So there are 4 important major aspects of the citric acid cycle in the exam point of view. The sequence of reactions, the energy producing sites, the inhibiting sites, the regulation sites. Okay, mention the enzymes, so concerned enzymes, that is important. Now the final thing, narrate the significance of citric acid cycle. In the significance, the first and foremost significance is the integration cycle of 3 metabolic pathways. Second significance, it acts as the producing cycle for the energy reducing equivalents like NADH2, FADH2, such reducing equivalents. Then third significance, it is amphibolic in nature. It is having both anabolic character and catabolic character. And fourth thing is anaplerotic, because it can replenish its own ingredients by interconversion of various compounds. So these are the significance of the citric acid cycle. So prepare it well. You have to draw the diagram. In all the textbooks, there are diagrams are given. So study it well. All the best.